very kind introduction. So it's a great pleasure to be here at the Data Science Institute to talk about my research or deep learning for images and graphs and how to apply these methods to solve problems in different areas of science so that there is a potential for collaboration with different research groups in chemistry, biology, material science, I think even chemical engineer, even physics. I will just give cover. So the intention here is I will cover quite a bit of my research and some of them are in detail. Some of them will be at a high level, so that if there is an opportunity to collaboration with any labs, so we can talk about a particular part in details offline. So talk about the deep learning. So this is one of the buzzword right now, hot area in machine learning, even in AI. But I actually started working on deep learning way before it became popular. That time I was still a PhD student. I did an internship with NEC Labs. So we worked on using deep learning for video analysis and action recognition. I think this is one of the earliest work on deep learning for video analysis. And so after that, I work on different areas of machine learning, deep learning, and solve problems in fundamental science, in biology, in physics, and in the medical domain. Today I will not talk about image recognition, and I will not talk about natural images and videos. So I will in particular talk about how to use deep learning to solve problems in fundamental basic science in cellular and molecular image. And I will not talk about the image recognition problem, but I will instead focus slightly different problem we call image transformation. And I will also move a little bit one step forward to talk about not necessarily grid-like data like images, video, text. I will talk about little bit about graphs. So these are extensions of a grid type of data, and I will show how can we use graph analysis techniques to solve problems, in particular at the molecular level. So so for biological image transformation problem, so we have a image here as an input. So we have, and our goal is to, we have a computational models in between, and our goal here is to transform the input image into different output images. So this is a very generic paradigm, and it has applications in different problem settings. One particular problem is image denoising. We know that in the field of microscopy, so when we try to take a image and take a picture of a certain biological tissue at high resolution, we either use light or electron microscopy. But in either case, we need to shoot a beam of laser or electron onto the tissue. So I think the trade-off here is if you shoot a very weak beam of laser or electron, so you will get a noisy image like the image in the upper part. But if you shoot a strong beam, that may somehow damage the tissue, and you will not get a faithful capture of the biological tissue. So the trade-off here is you have to somehow achieve a balance in between. And our goal here is if we have a noise image like the image at the top part, can we use deep learning AI to obtain a clear version of the image? And if we can do that, basically we can somehow solve this trade-off problem. So we can use hardware to obtain a noisy image, and we can use AI and software to transform that into a higher resolution image. And another example here is a structure, segmentation, and prediction. We know that if we are given a tissue, we want to visualize the structure, the biological structure inside the tissue. So the way to do that is usually we need to apply some staining to the tissue, and then the part of the tissue with that structure will show in certain color. So and if we want to, for example, if I have a piece of tissue, I want to show 10 different structures. Basically, we need to apply 10 different stainings, and we get 10 different colors. So obviously, there is a limitation that you cannot, for example, visualize. Currently, you cannot visualize more than, I think, 3 or 4 different structures, because each structure need to have a different color. So computationally, this there is a recent trend to solve this problem using computational techniques, basically given a unstained tissue. So we can use computational techniques to predict the structure inside the tissue. For example, here we show the structure of, I think, multiple different biological structure. That if we can solve this problem in terms of in using computational techniques, we can potentially identify many different structures inside the same piece of tissue. So this, if this is successful, we can essentially transcend the limit that can be achieved using experimental techniques. So these are examples of image transformation problems. So a common technique to solve this problem is using something called UNet. So this is a very well-known technique, and so the idea is quite intuitive. So basically, you have you can imagine here that you have an input image. So of course I show this as a one-dimensional. So if this is an image, it will be two-dimensional. So you have an input image, and there is an encoder path and there is a decoder path. So in the encoder path, you use either convolution or pooling techniques to gradually reduce the size of the feature map. Here, I think if it's an image, so the two-dimensional image will become smaller and smaller. And then this image will, eventually you have a quite small image. This will be used as input to the decoder. The decoder essentially is a reverse process. So you use transposed convolution and to basically or upsampling to convert this hidden code here into a larger and larger feature maps, and eventually you obtain a representation that is the same size as the input. Of course, you can transfer information directly from each level of the input encoder to the decoder by using skip connection. So this represents the skip connection. So this is a very well-known technique to solve image transformation problem. So you have an image as input, you have a image or feature map of the same size as output. And by training these models using matched pairs of input and output, basically you can figure out a parameter representation that capture the relationship between these paired training images. So one limitation of this unit is because the encoder and the decoder use either convolution or transposed convolution or pooling and unpooling operations. So all these operations are local operations in the sense that, so suppose this is the output, and this is the intermediate layer, this is the input. So each output unit here is determined by a small patch in the previous layer. And if you add, basically, this is, we call this receptive field. So, and if you increase the number of layers, so the receptive field, for example, for the output is somehow gradually increased. So if you add a lot of layers on top of each other, you get larger and larger receptive field. But this receptive field is completely determined by the network architecture. It basically depends on how many layers in total do you have, and what is the convolution kernel size in each layer. For example, if you use 3 by 3 convolution, for example, in each of these layers, basically the receptive field of this unit in this layer will be 3 by 3, and the receptive field size here will be 5 by 5. So all these are completely determined by the network structure, it cannot be changed once the network architecture is fixed. And it's always local unless you add a lot of different layers, and the receptor field become larger and larger. But in terms of each layer, the receptor field is still local. So what we have tried we have tried to do is try to come up with something that can be used to capture a larger receptor field, and the weight, etc. The network, the receptor field can be somehow determined automatically from the data. So what we have used is something called attention operator. So this is a very well-known technique these days, and if you have, for example, used Google Translator, etc. So basically, the underlying technique is the transformer network, and the attention is essentially the key of the transformer network. So in this attention operator, you basically have three sets of vectors as input called Q, K, and V. So what you try to compute is first, uh, you compute the similarity between Q and K. Okay, so you come up with a similarity matrix, and once you have a similarity matrix, you normalize the similarity in terms of each column, so that each column here sum to one. Okay, so once you have the normalized weight here, so you use this weight to combine the vectors in the V together. Okay, so by doing this, we get for each vector in Q, we basically obtain an output vector O here. So each output vector here is a linear combination of the columns of V, and the weight depends on the relationship between Q and K. So what is unique about this operator is the size of the output. The size of the number of vectors here depends on the number of vectors in Q. And each vector is actually a linear combination of the different columns of V. So you can see if we treat each output here as a unit, so each output here depends on all the vectors in V. And in some cases, this is attention operator. So there is a special case called the self-attention operator. In self-attention operator, we only have one input, one set of input vectors. This Q, K, and V are derived all based on the same input by doing a linear transformation. So basically, we have a one set of input vectors. By doing three different linear transformations, we get Q, K, and V. So that is called the self-attention operator. And if we generalize the self-attention operator to higher dimensions, basically we can apply that on images. For example, if we treat these vectors, okay, if we treat these vectors as input vector, and we apply self-attention operator, basically we can flat these dimensions so that we get a linear matrix. So each vector here become a vector in that as columns of a matrix, and we treat that matrix as the input to the attention operator. Okay, so by doing that, what we can achieve here is each output unit here will depend on all the input units, so that we can achieve a large receptive field in just one step. And this is very different. So sometimes it is confusing that people think this is very similar to the fully connected layer. Actually, it's very different. So a fundamental difference here is in fully connected layers, if you train your network or input of certain size, that size cannot be changed during prediction, because your size of your weight is determined by the size of your input. But here it's very different. So the size of your input can be very different from training and from prediction, because the weight here is not determined from the is not predetermined from the training data, but the weight here is dependent on the relationship between the different vectors. So this is how can we generalize the attention operator to high order data so that it can be used on data like images and graphs, videos, and even graphs. So So what we can do is this is the procedure. So we have a input tensor. Suppose our data, for example, is a three-dimensional data. So basically, we have an input tensor that is a four-way. We have a DHW. So this is the three spatial dimensions. C is the number of feature maps. So if, for example, if you are doing volumetric three-dimensional images, your input will be four-way because these three, the first three indices are for the three-dimensional XYZ of the image, and the C is the channel. So you may have multiple channels. So if you have a natural video, for example, you have RGB, C will be three here. So what we can do is this is how the self-attention operator works or high order data. So we apply one by one convolutions all these input tensor X. And these three convolutions are independent. So there are three different convolutions. By doing three different convolutions, we get query tensor, key tensor, and value tensor. These are three tensors. Each of them is also a four-way tensor. Okay, and once we obtain this is the self-attention operator. So we obtain the Q, K, and V, and we unfold these tensors, four-way tensors into each of them into two-dimensional matrices, Q, K, and V. So each of them here now become matrix. So you can see here by doing the unfold, we essentially treat the C, the channel as one dimension, the spatial dimensions all are collapsed. We all straight flatten the spatial dimension into one dimension. Okay, so this basically you have a matrix that it has many different columns. And so we use this QKV as an input to the attention operator. So this is essentially the attention operator. And after the attention operator, we get a matrix. Okay, so that and when we get output matrix Y, and we fold back the Y again into a four-way tensor. Okay, so this is the tensor, and the final output will be the tensor computed the basically the multiplication, sorry, the summation of x and y. So x is input tensor, y is computed by the attention operator, and the final output is the summation of these two. Basically, this is equivalent to that you have some four-way tensors as input. So this is the main path. We compute three different convolutions. We get Q, K, and V. These are the matrices, and we use these matrices as input to the attention, and we get output. And then this output is add together with the original input. So this is essentially the skip connection, and we get the final output. So by doing this, basically what we can achieve is something called size preserving global voxel transformer operator. So because by doing this operation, the size of the input will not be changed because the size of the input is h times w times the d. Size of the output here is again h times w times d. So what we so if we go back to the unit, basically we need, so this is how we achieve input and output of the same size, but in the unit, we need some operator, some layers which take input of certain size, but it either need to produce output as larger or smaller. In the encoder, we need to reduce the size, and in the decoder, we need to increase the size. And what we have showed is that we can basically achieve these two operations by doing some tricks here. So for example, this is the upsampling operator. So remember in the attention operator, the size of the output depends on only the query, only the queue, okay? Depends on only the size of the queue. So essentially, if we want to achieve a larger output, we basically need to uh, make the queue matrix to have more columns. So this can be achieved by some operation called transpose convolution with a stride of 2. So by doing a transpose a convolution of stride 2, so the size of the output here is increased by a factor of 2, so that if you apply, if you give this QKV as input to the attention operator, you get output that is increased. The size of this output is increased by a factor of 2. And similarly, you can produce output. Okay, you can produce output that is smaller than the input by reducing the size of the Q here. So you and the way to do that is you do a convolution here, but your stride is no longer 1. You can apply a stride of 2. So we know that if you do a convolution of stride 2, the size of your output will be reduced by a factor of 2. And if you reduce the size of the Q by a factor of 2, the output here is also reduced by a factor of 2. So by putting all these operations together, we basically have again this unit type of architecture. We have input here and we have output here, and in between is essentially an encoder and a decoder. So in the encoder, we apply this downsampling operator. Okay, so the size of the volumetric image is gradually decreased, and then this hidden representation is passed to the decoder, and the decoder will be essentially a reverse process. So each operation here will be an upsampling operator, and eventually we come back to a representation that is the same size as your input. Okay, and now we can here we can use loss functions, for example, cos entropy, etc., to train the network of matched pairs of images. So each of these downsampling operator could be the operator, our self-attention based operator we just described, and the operator in the decoder could be the operator that we just discussed. This could be one of the sampling operators. Of course, we have one layer here that is called size preserving. This one will not change the size of the feature maps. So we show that this network can be applied to do different types of image transformation problems. So one problem that we try to solve is called 3D image denoising. So in this data set, what we are given here is basically a very noisy input image. So this is just one example of image. So this is the input, and this is the last one is basically the higher resolution one. Okay, so this is the noise image. This is the clean image. You can see the difference is quite small, quite large. And this is these two are two zoom ins in these two locations. And if you use the unit in this paper, so that by using this natural method paper, so this is the output you can achieve. And this is the output you can achieve with our methods. And the last one is the ground truth. So this is what you can obtain if you actually do this using experimental techniques by shooting a stronger beam of laser. So quantitatively, we used the metrics used in the community called structure similarity and PSNR. So these are two different evaluation metrics, and the number higher is means better. So you can see this is three different conditions: C1, C0, C1, C2. So these are different level of noise or the input, and so this is the method using this natural method paper called CARE, and this is the metric that the value that we can achieve. And we also did the experiment to test how the size of the patch will basically affect the performance. So the idea here is we already said if you use UNet, so the size of your receptive field is essentially fixed. Regardless of what is the size of your patch, the receptive field is determined by the network. So that if you, the horizontal axis here represents different input size, you can see basically UNet will always achieve the same performance. So these three dashed lines are the UNet networks for three different conditions. And if you use our network, you can see with larger patch size, because in our network we can we can achieve a receptive field of the entire input patch in just one step. So if you give me in prediction time, if you give me a larger patch, and my receptive field will be larger. So this is essentially if you change the size of your input patch, and you can see the performance somehow is increased with a larger receptive field. We also applied our method of this 3D to 3D to 2D image projection problem. So the problem here is we have noisy 3D image of I think this is for the fruit fly. Okay, so the input is a three-dimensional image, and what we try to do is try to project the three-dimensional object onto 2D, because what we know that is in this uh, fly fly embryos. So all the important cells are on the surface of the embryo. So that my useful operation is we just need to project this surface onto 2D essentially, because we can essentially computationally unroll this surface area into onto a plane, so that we can better study the property. So what we try to do here is we try to use computational techniques to unroll project this three-dimensional object onto 2D, and this is what we can achieve use our method. And again, the baseline method here is in this CARE paper. So you can see in terms of structural similarity, our performance is actually much higher than if you use the plane unit. And these are some examples for the 3D to 2D projections. So this is the input. So this is what you can achieve using the unit method. This is what you can achieve using our method, and this is actually the ground truth, which is obtained using uh, experimental techniques. We also applied our technique a different data set. This is called the level-free prediction. This is essentially the problem that I described at the very beginning. Basically, we try to, given a unstained tissue, we try to predict the locations of different structures in this biological tissue without any kind of staining. We use pure computational to do it. These are the different names of these biological structures. And we used something called Pearson correlation. Basically, with Pearson correlation compute the correlation between our prediction and actually the ground truth. Of course, the higher will be better. So you can see our method achieved consistently higher Pearson correlation with the ground truth. And this is the difference, and this is the p-value. So you can see most of these p-values here are significant statistically. And these are some examples of this label-free prediction problem. Here is a different plot of the result. So this is a video show of this volumetric three-dimensional image. Here, this is each image here is a three-dimensional. So I show the z-axis as different frames of the video, so that we can see the entire three-dimensional volumetric image. So this is the input image, which is quite noisy, and this is what we can predict. So this is basically the output of our deep learning method, and this is the paired output that is obtained in using experimental technique, which we treat as a ground truth. So you can see here the prediction here from this very noisy input, we can achieve a prediction that is actually very close even to the ground truth here. So the last experiment we have done is try to test the idea of transfer learning, because we know that if we are given a problem to solve, this technique requires matched pairs of images. Basically, we need to have matched pairs of low resolution and high resolution images to train the network. And sometimes it may be hard to obtain matched pairs of images. So the idea here is uh, if I only have very small number or even no matched pair in a particular problem, can we use a model that is trained or matched pairs of different problem? Can we transfer that model directly into my domain? And if we can do that, we basically can minimize or even eliminate the need of obtain paired images for a new problem that we try to solve. So basically the idea here is I train a model or one of the data sets using matched pairs, and I directly use the trained model or a slightly different data set, how it's going to work. So what we have found is essentially that is the if we, for example, we tested our idea on three different data sets. So each row or each column here represent a data set. So you can see each row here, this is we train the model uh, of one particular data set. So each row corresponds to a different data set, and each column here represent a test on a data set. So this essentially we train the model on three different data sets, and we test all these models uh, each of the three data sets. We get this 9 by 9 configuration here. So naturally, you can see the diagonal part here represents that you train and test the model of the same data set. Okay, of course, this is we do train test split, but because the data is of the same nature, you can see the performance along the diagonal is naturally higher. And all the off diagonal ones represent the training and test happens on different data set. And what we can have observed is that essentially you can see there's in all of these off-diagonal settings, our attention-based method is much better than the convolution-based method in all of these settings. I think this is not this is consistent with the previous observation because attention-based models usually is more transferable than a convolution-based model because I think one guess is that because in the convolution you train the weight or the input, but you use all the training data, but you use the weight directly or the output or the target domain to make the prediction for the output. You transfer the weight directly, but in attention models, because the weight used to combine the input is determined by the relationship among the inputs. Now, so in that case, the weight or the training data are not directly transferred to the test data. So that is our work or you training this voxel transformers or matched pairs of data, and we also moved one step forward on the image domain because, as mentioned, so in many settings it will be very hard to obtain these matched pairs to train the network because our previous models are all based on supervised learning. Basically, you need to have these matched pairs to train the network. So and we move one step forward by asking ourselves, can we use unsupervised learning techniques to train the model or not? So what we have used is something called self-supervised learning. So this is one of the cutting edge research areas in deep learning. So if we have a model, can you use a type of unsupervised learning to train the model or not? So the self-supervised learning is basically you use one part of the, for example, if you try to train an image model, basically you can use the one part of the image as input and try to predict other part of the image. You can use the left half as input to try to predict the right half as output. By doing that, basically you are able to train a network so that the network can capture some kind of statistic in the training data. So you can use the past to predict the future. You can use you can mask one part of the input and try to use the other part to predict this part of the input. And we developed this method called noise to save. Basically, you have a input as a noise image. We try to learn from itself, and we try to produce a clear version of the image. So this is one of the new RIPS paper last year we published, and this is quite a technique, but what we have derived is basically we can derive self-supervised upper bound or the supervised loss. So in the supervised training, matched paired training, we basically train this supervised loss. But if you don't have Y, if you don't have Y is basically the output that you obtain using experiment. If you don't have Y, we show that uh, we can obtain an upper bound or the supervised loss. We call this is a self-supervised loss because you don't see a Y here. This is all based on the same noisy input. And
Shuiwang,
yes,
can I ask you a question here?
yes, please.
So here, do you, when you get this result, do you assume this the image, if you have multiple image, like you have image in the sequence on your previous slides, you have this kind of time, right, dimension. Do you assume those kind of sequence of images should be also independent from each other?
Uh, in the previous slide, we showed this image is we showed image as a time series, but actually it's not. It's a volumetric three-dimensional image. So
So it's not like a video is a continuous the image stream, one image.
right, it's not a video. It's basically because it's the biological tissue is a three-dimensional, so you get a volumetric three-dimensional image. But what I showed there is these slides is shown as a video frame just for visualization.
Okay, thank you.
And here, the same technique, this can be applied to volumetric images as well. It doesn't assume that the image has to be 1D or 2D or 3D. Actually, we apply this to the image, the biological image in the previous experiment. So this is a quantitative results. We tested our model on images from very different areas. So these are the image net, and this is Chinese characters called Hanzi, and this is basically the biological image that we used in previous experiment. And um, you can see in terms of performance. So these are the different each row here represents a different method. So these three methods is essentially the traditional method. These two are the supervised method if you really have the matched pairs. And these are the self-supervised method. So this is the result of natural images. I think this is one of the widely used image data set for denoising. So this is your first column here is your input. So this is each column here represents output of different methods, and this is ground truth. So and these two basically are supervised method. So noise to true here represent the supervised method. You really have the ground truth, and this is the result of our method here. So we also use apply this on the biological image data set, and the setting there is the same. Basically, we have an input. This is the noisy input. So the last column is the ground truth, and this is what you do if you use a supervised training. Okay, and this is if you use our method, which is unsupervised. So you can see the each row here represents different condition, which means different noise level. You can see if the noise level is very large, okay? So if the image is very noisy, you can see our method is also is not very similar to the ground truth, but still I think it's better than other self-supervised method. Okay, so that is uh, about my part of work or images. So next I will move a little bit faster on the graph-based data. So what we have been doing in the past around 3 years, we try to extend our method on this grid type of data to more generic data. For example, if we work on images, basically you can consider image as this 2D pixel, the pixels lie on a regular lattice of the 2D. And if you are dealing with either text or speech data. Basically, you are doing a one-dimensional time sequence, time series data. So what you have tried to extend is if we treat the image essentially can be considered as a special case of a graph, and how can we extend this method to more generic graphs? In graphs, these nodes may not lie on regular grid, and each node here, each node can have different number of neighbors, and the neighbors has no order. So that's very different with images because images, if you don't look at the images on the border, basically each pixel here has same number of neighbors, and the neighbors are nicely ordered in certain way. But none of this is true for graphs. So we try to extend these methods of graphs and use this to solve problems in different domains, especially for drug and material science. Because if you have basically if you have graph, you only have these nodes and this relationship, and nodes and the object and the relationship together basically give you the graph information. So and the graph is actually not new for machine learning and data mining and AI. So we can use graphs to solve different problems. So these are the typical problem. We try to do node classification. So in this case, each node here is an object. We try to predict a label for node. And over here can also do graph classification. In that case, we try to predict a label for the entire graph. And we also can do link prediction. We try to predict if there will be a link between two different nodes. And of course, we can do community detection. So for example, here, if we have a citation network, each node here can represent a research publication, and the edges here represent the citations among these publications. And we can try to, for example, predict a category for each of the publications. For example, this one could be a computer science publication, the other one could be a math, etc. So this is an example of document network, and this can be formulated as a node classification problem. We can also uh, formulate, for example, this molecular property prediction problem as a graph classification problem. So the task here is we are given a molecule as a graph, and we try to predict the property of this molecule. For example, if this molecule is toxic or not toxic, if the molecule can be used as a drug or not. So I will talk about a little bit more about this. So um, one of the research team in my team here is we try to use treat molecules as some kind of special graphs, and we try to either to do molecular property prediction in different domains, for example, in quantum mechanics, in physics, in chemistry, etc. And we also can essentially design graphs. We generate graphs based on some training data. So for the sake of time, I think I will move a little bit faster. So one recent work that we have done is something called 3D graphs, because when you deal with, for example, biological data, molecular data, so I think the typical practice is to treat each molecule basically as a graph. But the problem with that approach is basically the location information of the nodes has been lost, because if you treat a molecule as a typical graph, the relationship between the nodes has been captured, but the location of the nodes has been lost during that time. Because if your molecule basically is a 3D object, each atom in the network has a special coordinate, and it's actually useful because if, for example, if you try to predict a molecule can be used as a drug or not, the geometry of that molecule is very important because it need to fit into the pocket of the target protein. So the existing approaches in the graph domain, graph neural networks, only consider partial geometry. For example, this SCHNet only consider the distance among the atoms, and the more recent work called DimeNet only consider the pairs of angle between pairs of chemical bonds. So what we come up with is we studied the problem in depth, and we actually figure out that there is a additional degree of freedom that need to be fixed. If you only capture, for example, one angle, you cannot uniquely determine the location of the atom. So this is an example that we found out. And to solve that problem, we come up with this unified framework for 3D graphs. And the paper came out just recently, and I think it's all archived. So if you are interested, you can look at the paper. But here, what we try to do here is we first, based on a unified framework developed by DeepMind in this paper, I think this is about 3 years ago, they developed a unified framework for message passing in graph neural networks, and we systematically extended that framework to include location information. We call this three-dimensional graph networks. And based on this um, framework, we developed something called spherical message passing. So we define, so this is based on the quantum chemistry in the spherical coordinate. So we define we define the location of each node by using the spherical coordinate. Basically, you have a radius. So this is distance to the origin. Then you have two angles. One is called theta, the other is called phi. And we can show that if you use these three to define the locations of each atom, the location can be uniquely determined. And there is quite a bit of techniques in this paper. How can we use quantum chemistry, Schrodinger equation, etc., to compute a precise representation for our molecular representation? And we show that if we use this our technique to solve problems in quantum mechanics and in the chemistry. So this is one of the quantum chemistry problem to predict the energy level of catalysis. So we can see that if you can incorporate complete geometric information, you can obtain consistently better performance. Okay, so that is about our 3D network, and we also worked a little bit on something called explainability, because when you try to make predictions, and deep learning these days are criticized as a black box. It can make accurate predictions, but the results are very hard to be interpreted. And what we try to do is we try to interpret the results of graph neural networks. So there is already prior work on explainability, but I think this work, they try to identify individual nodes, individual edges that are important for the prediction. So the key difference of our work is we think that if you identify nodes or edges that scatter around the graph, it will be very hard for us to interpret. But rather what we did is we intend to identify just a subgraph. For example, in this all this sub input graph, we try to identify a small subgraph, and by using only this subgraph, we already can make accurate predictions instead of the entire graph. And by doing that, we essentially show that this is essentially the key part of the graph that is responsible for the prediction. So we develop a multi-color tree search-based method coupled with a game theoretical scoring function to guide our heuristic search. And there will be a lot of details in the paper posted on archive very recently. But I will skip the details, but just show you some results we obtained. So you can see here, this is the result we obtained. So this is synthetic data used in by the community, and we know that this part of the graph is actually the motifs that is used to distinguish a different category. You can see if you use our method, you can accurately identify this part. But if you use other method, somehow you can identify this structure, but you also have other noise information that is identified. And this is show if we how can we evaluate our method using a metric called fidelity. Okay, so of course, fidelity means higher or better, and if you change the prediction, basically it measures change of prediction if you remove the important substructure. And you can see here our fidelity value is much higher than other previous methods. Okay. So I think I only have a few minutes. I will talk a little bit about the last part of my work, which is graph generation. So here, the fundamental goal here is if we try to use computational methods to generate graphs. In particular, we try to generate molecular graphs with certain property, because in the drug design industry, we try to come up with a molecule that has certain property. For example, it will inhibit certain bacteria, and it can be used as an antibiotic, etc. But to synthesize this is quite expensive. So what we try to do is, we given a set of molecules with certain property, we try to generate new molecules with certain desirable property. So this is the molecular graph generation problem. So given a set of molecules, so we try to train a generative model, and the model will capture the distribution of this input graph. And once the model is trained, we can use this model to generate a new molecule that is not in the training data. And we can also do property optimization. So this is related to the first one, but slightly different, because if we have, for example, you have these molecules, and we know the property values, these values, we can use this to train a model, and we can optimize the model so that the model can generate molecules with desirable property values. For example, if we want to generate a molecule with higher um, property value, we can try to tune the model by doing this as a reinforcement learning problem, and we can optimize the model so that it can generate molecules with higher or lower desirable property values. So we recently we developed this model called the discrete flow model. I don't think I will go into the details, but because the key difference of our work with the previous work is because graph generation is basically a discrete generation problem. We generate a node, we need to know the type of node. When we generate edge, we need to know the discrete type of edge. Basically, this is a discrete problem. And our previous work used a continuous latent variable model to do the work. And we can show that their method cannot capture the discrete distribution faithfully. And we developed this discrete normalizing flow model for graph generation recently. And by using this method, we can also do property optimization by using the reinforcement learning paradigm. Basically, we can treat this as a Markov decision process. We have state, action, reward defined, and because when we generate a graph, basically there are multiple steps, and the final value will only come if the molecular graph is completely generated. So each step of generating a node or edge will be treated as one action, but the reward only come after the molecule is completely generated. So we treat this as a reinforcement learning problem. We also try to develop a energy-based learning method to do molecular graph generation. So this is basically an extension of a probabilistic model. So the what we have done is we try to, so the energy-based learning is quite old area, but it has been used to do image generation, image modeling task, because images can be considered as continuous. And we try to make this to work on graphs recently. So this is some results of molecular graph generation using our discrete flow model. So the upper part here shows the results of unconditional generation. Basically, we will just want to generate some molecules which might, and the value, the matrix here are based on the validity, uniqueness, one novelty, and the reconstruct. So this is the unconditional generation, and the lower part is the conditional generation. Basically, we call this a goal-directed generation. We want to generate a molecule with certain desirable higher property value. And you can see that if you use our discrete flow model, you can generate molecules with higher desirable values. And this is a result of using energy-based models. So I want to, in particular, show that one very unique property of this energy-based learning is we can do multi-objective generation. So, for example, if you want to generate a molecule with two properties, two higher values for two properties. So in the previous method, you can only optimize one property value. If I only, for example, I want to my molecule to have a low toxicity, low toxic, and also I want my, for example, molecule to have higher energy, for example. So there, in previous method, you cannot do this, but our energy-based learning method can naturally do multi-objective generation. So this is we tested these two different properties, and you can see this is the projection of this property onto the two axes. You can see our method can achieve higher value in each of these two properties if you uh, project the data onto either axis. So lastly, in addition to publishing our work, we also recently tried to put all these uh, our method together, try to develop open source uh, software library called Dive into Graphs. So what we have found is whenever you do a research in different areas of graph, basically there is a lot of work uh, to compare, come up with data matrix and compare with different prior methods, and that consume a lot of time. So what we try to do here is we try to incorporate all of these implementations, data set evaluations, etc., into one place. And we try to, and currently we are working on these four areas of graph neural networks. And if you are working on any area of this research, and if you grab our code, you can basically reproduce the results of all previous methods, I think within a couple of hours. And then you can start testing your new ideas, compare with baseline quite easily. And if you right now there is no such a turnkey solution to test your method, and that is essentially our goal. And the toolkit is not ready yet. I think it will come probably in a few weeks. Okay. So briefly to conclude, we one of our goal is to use large-scale concerted efforts of the entire group to solve challenging problems in different areas. And currently, our research involve on one hand involve developing fundamental computational methodology. On the other hand, we try to solve fundamental scientific problems in biology, neuroscience, and chemistry, and physics so far. And most of our problems are focusing on micro scale problems at cellular, molecular, and we are probably moving to the atomic world of atoms and atomic levels right now. So finally, this is teamwork and involve a large number of students. Three of the students that are involved in this work have graduated last year. So they either go to industry or academia, and there are also quite a few PhD students and actually one very good undergraduate student working with us on this active research projects. Okay, I think